Dear friends, from last lecture we have discussing the most important topic from NEET point of view, that is the excretory product and their elimination according to CBSE pattern. After completion of basic concept regarding the excretion and osmoregulation, we have discussed about the what is meant by excretion, different modes of excretion in different animals. We have prepared the chart which showing the different animals and their excretory organs and now we want to go in details of human excretory system, because you know that in human being excretory organ, that is the metanephric kidney. Now concentrate here, that is the human excretory system. When you focus on this human excretory system, this human excretory system made up by the several organs which connected with each other and form the system, that is human excretory system, which plays important role, that is the excretion and osmoregulation. Now this excretory system consists of several components. That is first, that is the a pair of kidneys, then a pair of ureters, then third, that is urinary bladder, and fourth, that is urethra. So there are four components: a pair of kidneys, pair of ureters, urinary bladder and urethra. Now we want to go in details of each. Now concentrate, this is the pair of kidneys, this is the pair of ureter, urinary bladder and urethra. So these are the different part. Now first concentrate on the pair of kidneys. When we focus on these kidneys, generally kidneys are somewhat bean shaped. Kidneys are bean shaped, and these bean shaped kidneys located in the abdominal cavity, at dorsal side of the abdominal cavity. So the position, it lies between second thoracic, means here it lies between, that is 12th thoracic vertebrae to the third lumbar vertebrae. So the exact position, it lies at a dorsolateral side of 12th thoracic vertebrae to the third lumbar vertebrae. Average dimension of that is the kidney, that is average length, it is 10 centimeter, average width it is 6 centimeter, while thickness it is about 4 centimeter. Each kidney weighs about, average weight it is 135 to 140 gram is the average weight of each kidney. When we observe the superior pole of kidney, there is presence of a cap-like structure, and this cap-like structure is termed as a adrenal gland. It is an endocrine gland. This adrenal gland also termed as a suprarenal gland. If you observe this both kidneys, generally this right kidney is slightly lower than the left kidney. It is the peculiarity, because right side consists the liver, and due to presence of liver on right side, right kidney is slightly lower than the left kidney. These kidneys are bean shaped, so it shows two regions. This is the concave region and this is the convex region. This concave region shows the depression or opening termed as a hilum or hilus. It shows as hilum or hilus, through which renal artery enters into the kidney, while renal veins goes out from the kidney along with the ureter. So hilum or hilus is the conical shaped depression present at concave side of kidney, through which renal artery and nerves enters into the kidney, while renal vein and ureter leaves the kidney. Generally these kidneys are innervated by, means dorsal aorta from the heart brings the oxygenated blood, and this oxygenated blood, which is impure blood, which supplied to the kidney is the organ of purification of blood. All impure components from the blood get removed from the kidney, and pure blood released as a deoxygenated blood, but deoxygenated blood released into the renal vein, and from renal vein arises the inferior vena cava. This inferior vena cava send this blood towards the heart. So dorsal aorta supply oxygenated and impure blood towards the kidney, while the inferior vena cava carries the pure blood towards the heart, which is deoxygenated. It is most important. Sometimes we are making the mistake, that is the impure and pure blood, oxygenated and deoxygenated blood. Oxygenation and deoxygenation, oxygenation process occurs in the lungs, while the purification of blood occurs in the kidney. So kidney is the organ of purification of blood. Okay, now after kidney, next part, that is the ureter. When we focus on the ureter, ureter is a long muscular tube, measures about 25 to 30 centimeter. Measures about 25 to 30 centimeter. It starts from the hilus region or concave side of the kidney up to the runs through the pelvic region and opens into the urinary bladder. This ureter opens urinary bladder and it have the oblique opening. So ureter opens into urinary bladder through oblique opening. The significance of oblique opening prevents the transfer of urine from urinary bladder towards the kidney during it get filled. So it is the significance of oblique opening of ureter from posterior side into the urinary bladder. This ureter lined by pseudo, means here it is, when we focus on this, it is lined by this epithelial cells, and these epithelial cells mainly involved in the transfer of urine from kidney to urinary bladder. So function performed by ureter, it transfer the urine from kidney to urinary bladder by peristalsis. Peristalsis, rhythmic contraction and relaxation of smooth muscles. Now third component of excretory system, that is urinary bladder. Urinary bladder, it is a thick muscular inverted pear shaped sac lies in pelvic region or sacral region, and this muscular bag, which is pear shaped, lies in pelvic region, consists two components, that is the outer peripheral part is body while central part is trigone. This outer peripheral part body consists a special type of muscles termed as a detressor muscles. Special type of muscles present here, it is termed as a detressor muscles, which brings the contraction and relaxation during micturition, while central part, which is the hollow part, and this central part is termed as a trigone, and this trigone central part lined by special epithelium termed as a transitional epithelium. So generally when we focus on the anterior region, that is the pelvis, ureter and urinary bladder lined by transitional epithelium. This is most important aspect, that is what is meant by transitional epithelium. Transitional epithelium, it is similar to this stratified epithelium, but difference between stratified epithelium and transitional epithelium, in stratified epithelium we observed the basal layer, that is stratum germinativum, and this stratum germinativum was absent in the transitional epithelium. Significance of this transitional epithelium, it have ability to stretch, and due to this stretching ability, the diameter of the diameter of the urinary bladder increases, and it receives the maximum amount of urine, because it is temporary storehouse of the urine. So here it is, main function performed by urinary bladder, that is temporary storage of urine, and remember normal volume, it is about 300 to 500 ml. Normal amount of urine stored into urinary bladder, it is 300 to 500 ml, but in certain condition due to the transitional epithelium, it can extend, and maximum it is 1 liter to 1200 ml urine will be stored into urinary bladder in certain extreme conditions. At the junction of urinary, next organ, that is the urethra. This, when we focus on the urethra is the small duct which release urine outside the body from urinary bladder. So urethra, it is the duct, and this duct eliminates the urine outside the body from urinary bladder. The length of urethra varies in male and female. In female, this urethra is have about 4 centimeter in length, while in male, generally in erectile condition, it have average length, it is 18 to 20 centimeter. When we focus on this urethra in male, it helpful in the elimination of urine as well as it discharge out the semen during the insemination process, or that is the insemination process. So it is also termed as urinogenital duct. It is also termed as urinogenital duct, while in female it is exclusively for urine elimination. So this is the urethra. At the junction of urinary bladder and urethra, we can observe the urethral sphincters. There are two types of urethral sphincter, that is internal urethral sphincter, and second, that is external urethral sphincter. So sphincters present at the junction of urinary bladder and urethra, there are two types, internal sphincter and external sphincter. Now when we differentiate these two, internal sphincter, it is involuntary in nature, while external sphincter, it is voluntary in nature. And this urethral sphincter plays an important role in the event, that is micturition event. So this is about the human excretory system. From neat point of view, prepare a flow chart. Take in column 1 different organs, in column 2 their peculiarities, and in column 3 the function performed by these organs, and make a mention in a tabular form. In theory exam, prepare in detail structure with the help of diagram, describe the human excretory system. So describe these 4 different components of the human excretory system in briefly. Okay, this is about human excretory system. So once again, a pair of kidneys mainly involved in the process that termed as a excretion and osmoregulation. Other functions also performed by the kidney to maintain the acid base balance. It regulates the pH. In some time it secretes the hormones. These are also functions performed by the kidney, but two important functions it involved in the excretion and osmoregulation. The ureter is a tube which transfers the urine from kidney to urinary bladder. Then urinary bladder, it is a temporary stores the urine. And last, that is urethra is duct which eliminates the urine out from urinary bladder to outside. At the junction of urinary bladder and urethra, we can observe the urethral sphincter, which is of two type, internal and external, helpful in the event, that is micturition. What is micturition? It is voluntary act of urination is termed as micturition. Now after completion of human excretory system, we want to focus on the detailed aspects of human kidney, that is histological structure of kidney. Generally in board examination, it is the most important aspect. Generally after every 2 exams, this structure will be repeated as a describe the histological structure of kidney, or draw a neat level diagram of VS of kidney. In neat examination, we want to focus on the detailed aspects present in this kidney. When we focus on the VS of kidney or histological structure of kidney, these kidneys are externally covered by a covering, and this covering, external covering, have the 3 layers, that is renal fascia, it is the outer layer, renal adipose capsule, it is the middle layer, while renal capsule, it is termed as the inner layer. So outer, middle and inner. Outer layer is termed as the renal fascia. Outer layer is renal fascia, and this renal fascia, it is thick layer made up of fibrous connective tissue, and main function, that is the protection. Below this renal fascia, there is presence of a fatty tissue layer, and this fatty tissue layer, it is made up by the termed as the adipose capsule, and this adipose capsule acts as a shock absorber. absorber, while the third layer, that is termed as a renal capsule. Renal capsule is the innermost layer. It is the innermost layer, and this innermost layer is the thin layer of again fibrous connective tissue, thin layer of fibrous connective tissue. And most important function, it separates out the kidney from ureter and prevents the kidney from the infection, So this is the function performed by renal capsule. So the covering of kidney have the three layers, that is renal fascia, outer covering, middle is adipose capsule, and inner, that is the renal capsule. Renal fascia is made up by the fibrous connective tissue, perform function protection. Adipose capsule made up of fatty tissue, shock absorber, and renal capsule is the innermost thin layer of fibrous connective tissue, separate it from the ureter, and it prevents the infection in kidneys. So when we focus on this VS, internally kidney shows two distinct regions, that is outer few mm, means 2 to 3 mm region is termed as a renal cortex, while inner region is termed as a renal medulla. So these are the two components, that is first is termed as a renal cortex and second is termed as a renal medulla. When we differentiate the renal cortex, this renal cortex, it is granular in appearance and red in color. Granular in appearance and red in color. Red in color due to presence of blood capillaries. So it is red in color, while granular appearance because it consists Malpighian bodies of the nephrons. So it consists the Malpighian bodies of the nephron, and due to this Malpighian body, it shows granular appearance. So renal cortex is red in color and granular in appearance. Red color due to the presence of blood capillaries, while granular in appearance due to presence of Malpighian bodies or Malpighian corpuscles into the renal cortex. Now second, that is the renal medulla. Renal medulla is the inner large area of the kidney is termed as a renal medulla. This renal medulla consists 8 to 16 conical shaped pyramids. 8 to 16 conical shaped pyramids, and this conical shaped pyramid consists the loop of Henle and collecting duct of the nephrons. It consists loop of Henle and collecting duct of the nephron, because nephron or uriniferous tubule is the structural and functional unit of kidney. Each kidney consists 1.2 million nephrons, means average there are 12 lakh nephrons are present, and these nephrons consist the different components in which there is loop of Henle and collecting duct. This loop of Henle and collecting duct shows striation appearance, so it is termed as a striated in appearance and light red in color because it does not consist the blood. So here it consists the renal filtrate. It consists the renal filtrate, and that's why it is striated in appearance and lighter in color. Renal medulla is striated in appearance and lighter in color. In renal medulla, we can observe the different pyramids, which are conical shaped pyramids. See here, this is the conical shaped pyramids, and these conical shaped pyramids shows two regions. The broad region of this pyramid, it is towards cortex side, while the narrow apex towards pelvis side. So broad surface towards cortex side, while narrow apex towards the pelvis side. So it is a conical in shape. This narrow inner side have the opening termed as a renal papilla. Plural form, it is termed as a renal papillae. So at the base of each renal pyramid there is presence of renal papilla. Together it form the renal papillae. Now each pyramid gets separated from another pyramid by a column of Bertini. So renal column of Bertini is the extended portion of the cortex lies in the medulla and separate out the pyramids from each other, termed as a renal column of Bertini. Now each pyramid opens into a small duct termed as a minor calyx, but 3 to 4 calyx group together and forms the minor calyces. This is one calycis, this is second calycis, this is third, and these three minor calyces opens into calyx termed as a major calyx. Major calyx, 2 to 3 major calyx calyces opens into the pelvis, So in this way there is formation of pelvis. Now what is pelvis? Renal pelvis is an anterior funnel shaped portion. This looks here, anterior funnel shaped portion, like this, anterior funnel shaped portion which collect the urine which is prepared into the uriniferous tubule or nephron. So ureter is the anterior funnel shaped portion of ureter present in the renal medulla, which is lined by transitional epithelium, and from this renal pyramid arises the ureter, while renal artery and renal veins are associated with this histological structure. So it is the histological structure of kidney or vs of kidney. Once again remember, entire structure is covered by covering, and this renal covering is made up by 3 layers: renal fascia, adipose capsule and renal capsule. Histologically, kidney shows 2 distinct regions. Outer peripheral part is cortex and middle part is termed as a renal medulla. Renal medulla consists 8 to 16 conical shaped pyramids. These conical shaped pyramids separated from each other by renal column of Bertini. These all at the base of each renal pyramids we can observe the renal papilla. Renal papilla joins together and opens into the small duct. It is minor calyx. Minor calyx opens into the major calyx and major calyx opens into the pelvis. So this is the histological structure of the kidney. After human excretory system and histological structure of kidney, we want to focus on the most important aspect, that is the nephron or uriniferous tubules. This nephron or uriniferous tubule is the structural and functional unit of kidney or excretory system. So nephron is the structural and functional unit of kidney or excretory system. Average number, that is each kidney consists 1 to 1.2 million nephrons. 1 to 1.2 million nephrons. Remember, from exam point of view, that is 1.2 million nephrons. So sometime question asks, both kidney consist the nephrons, that is 24 lakhs, means 1 million is equal to 10 lakhs. So both kidney consist 2.4 millions, means it is about 24 lakhs. So average number of nephrons present in each kidney, it is 1.2 million. The average length, it is 3 to 6 centimeter is the average length of nephron, while average diameter, it is 30 to 40 micrometer is the average diameter, but this diameter varies from the several regions of the nephron, When we focus on this nephron, total nephrons present in each kidney, these nephrons are of 2 types. That is, first, it is termed as cortical nephrons, and second, it is juxtamedullary nephrons. Cortical nephrons and juxtamedullary nephron, means out of total number of nephrons present in kidney, there are 2 types of nephron, cortical nephron and juxtamedullary nephron. Generally 80 to 85% nephrons are of cortical nephrons, while the 15 to 20% nephrons are juxtamedullary nephrons by number. The most important property of cortical nephron, in cortical nephron, Malpighian body, it is Malpighian body lies in cortex region of kidney, cortex region of kidney. During regular lecture you have studied what is Malpighian body, different part. We want to focus also in this lecture on the base in detail from NEET point of view. So Malpighian bodies of cortical nephrons lies in cortex region, while Malpighian body lies of juxtamedullary nephrons lies at the junction of cortex and medulla, at the junction of cortex and medulla. This is the property. Next, that is the in cortical nephron we can observe diameter difference between afferent arteriole and efferent arteriole. Afferent arteriole and efferent arteriole means afferent arteriole have bigger diameter than efferent arteriole, while in juxtamedullary nephron no diameter difference, means both arteriole have the same diameter, both arterioles have the same diameter. Then in cortical nephron, that is loop of Henle is small or short in size, while in juxtamedullary nephron loop of Henle is long. So this is in cortical nephron, distal convoluted tubule lies away from glomerulus, while juxtamedullary nephron, in juxtamedullary nephron DCT associated with glomerulus and forms JG apparatus So JG apparatus is the peculiarity of juxtamedullary nephron, while cortical nephron does not have JG apparatus, does not consist the JG apparatus. So peculiarity of JG apparatus, juxtaglomerular apparatus, is the peculiarity of juxtamedullary nephron, while cortical nephron does not consist the juxtaglomerular apparatus. This cortical nephron works during normal physiological condition. So during normal physiological conditions, this cortical nephron works, while juxtamedullary nephron works during stress conditions. When water level in blood decreases, when sodium level in blood decreases, this juxtamedullary nephron works. So this is the difference between cortical nephron and juxtamedullary nephron. Means in kidney we can observe the two types of nephron, that is cortical nephron and juxtamedullary nephron. From regular point of view, differentiate cortical nephron from juxtamedullary nephron, and from NEET point of view also we want to focus on the peculiarity of nephrons. In general general pattern we are focusing on the cortical nephron, not juxtamedullary, but for the regulation of kidney function we need a juxtamedullary nephron. जेव्हा आपल्याला डिफरन्शिएटिंग पॉइंट्स विचारले जातील तेव्हा 6 पॉइंट्स तयार होणं गरजेचे आहेत, कारण का नेहमी डिस्टिंग्विश पॉइंट हे 2 मार्क्ससाठी विचारतात, तेव्हा आपल्याला त्याकडे 4 पॉइंट्स लिहिले की 2 मार्क्स मिळतील, पण कलेक्शन जे 6 ते 7 पॉइंट्स असायला पाहिजे. म्हणून डिफरन्स काय तयार होतोय, आउट ऑफ टोटल नेफ्रॉन 80 to 85% nephrons are cortical nephron, 15 to 20% nephrons are the juxtamedullary nephron. First point. Second, in cortical nephron Malpighian body lies in the cortex region of kidney, while in Malpighian body of the juxtamedullary nephron lies at the junction of cortex and medulla. Third point, there is a diameter difference between afferent arteriole and efferent arteriole. In juxtamedullary nephron, there is no diameter difference between afferent and efferent arteriole. In cortical nephron, loop of Henle is short in size, while in juxtamedullary nephron, loop of Henle is long. In cortical nephron, DCT, distal convoluted tubules, lies away from the glomerulus, while in juxtamedullary nephron, DCT is associated with the glomerulus and forms the apparatus, that is JG apparatus. So, presence of JG apparatus is the peculiarity of juxtamedullary nephron, while absence of JG apparatus is the peculiarity of cortical nephrons. And function, generally normal physiological conditions, cortical nephron works in the kidney, while during stress condition or adverse condition, juxtamedullary nephrons works generally when decrease in body water content in the body fluid, and second, that is the decrease in sodium level in the blood or body fluid, then it works, that is juxtamedullary nephron. So, this is about the nephron. Now, we want to go in the details of nephron or uriniferous tubule from NEET point of view.